In this video, I will be discussing the Hershey and Chase experiment. Conducted in 1952, the purpose of this experiment was to determine whether proteins or DNA are responsible for transferring genetic material. In order to test this, Hershey and Chase decided to track the transfer of protein and DNA between a virus and a host. The experiment used T2 bacteriophage as the vehicle for delivering genetic material. The simplicity of the virus, which contains a protective protein coat and a DNA core, made the perfect vehicle. The host used was E. coli. Two experiments were conducted. The first tagged the T2 phage DNA with radioactive phosphorus, phosphorus 32. This was used because DNA contains large amounts of phosphorus, and the phosphorus 32 allows the researchers to track the DNA throughout the transfer process. The phages were then allowed to infect the E. coli. After the phage was introduced to the E. coli, a Waring blender was used to detach the protein shells of the virus from the E. coli cell. A centrifuge was then used to separate the bacterium from the phages and the protein. After the separation was complete, the radiation concentrations were measured. The experimenters found large quantities of phosphorus in the bacterial sample, which showed that DNA was transferred to the host organism, while also still remaining in the virus. The experiment was replicated, but this time phosphorus 32 was replaced with sulfur 35 because it appears in the amino acids that make up proteins. Sulfur is not present in DNA. The result of this experiment showed only radioactivity in the protein shells and not in the host. The conclusion of Hershey and Chase's experiment was that DNA, not protein, is the genetic material. The experiment helped convince the world that DNA was genetic material.